In this video tutorial, I am going to discuss about regulation of urea cycle. In earlier video, we had seen the steps or reactions of urea cycle. Here we will see that how urea cycle is regulated. Actually, there are two levels of regulation for the urea cycle. One is short-term regulation and second one is the long-term regulation. So let's first look at the short-term regulation of urea cycle. In the earlier video, we had seen that urea cycle is a 5-step process. Out of this 5 steps, this first step is unique. Why? Because this first step in the urea cycle, it is a rate limiting as well as committed step of urea cycle. Now, what is the meaning of rate limiting step? See, in this entire urea cycle, this first reaction is the slowest reaction. So what happens? The speed of this reaction, it determines the speed or rate of entire urea cycle. That's why we can call it as a rate limiting step. The second is committed step. What is the meaning of that? See, in this first reaction, this carbamoyl phosphate is synthesized inside the mitochondria. And this carbamoyl phosphate, it has only one fate, that is to enter in the urea cycle. So we can say that once this first reaction takes place, then there is a commitment towards the urea cycle. That's why it is called as a committed step also. Now, in any metabolic pathway, when particular reaction is either rate limiting or committed step, that becomes very good target for various regulatory molecules. Similarly, here also carbamoyl phosphate synthetase 1 is a good target for various regulatory mechanism in our body. Now, carbamoyl phosphate synthetase 1 is under a positive allosteric influence of one molecule, that is N-acetyl glutamate. This N-acetyl glutamate, it is often abbreviated as a NAG. Now, this N-acetyl glutamate is actually obligatory or a compulsory requirement by this carbamoyl phosphate synthetase 1. That means if N-acetyl glutamate is absent, then carbamoyl phosphate synthetase 1 cannot act, right? Now, how N-acetyl glutamate is formed? N-acetyl glutamate, as you can easily imagine, it is synthesized from the glutamate and acetyl coenzyme A. So, glutamate plus acetyl coenzyme A. Now, here what happens? Acetyl portion of acetyl coenzyme A, it binds with the glutamate to give rise to N-acetyl glutamate. And here the enzyme is N-acetyl glutamate synthase. Now, once N-acetyl glutamate formed, it activates the urea cycle, and when the function of N-acetyl glutamate is over, it is broken down to glutamate and acetate. And this breaking down reaction is catalyzed by the N-acetyl glutamate hydrolase. Now, here N-acetyl glutamate synthase is under a positive allosteric influence of asparagine. So, as we can see over here, asparagine, it stimulates N-acetyl glutamate synthase, which in turn increase the synthesis of N-acetyl glutamate, and then it stimulates carbamoyl phosphate synthetase 1, so urea cycle gets activated, right? So, this is the short term regulation of the urea cycle. Now, let us look at the long term regulation. Long term regulation of urea cycle. But before we discuss the minute detail of long-term regulation of urea cycle, there is one observed effect, and that is that whenever there is increased amino acid level for the long time, it is observed that in such condition it stimulates the synthesis of all enzymes of urea cycle. Now, this is just observation how exactly increase amino acid in the blood for the long time, it stimulates synthesis of all enzymes of urea cycle, that is not known. Simply this is the observation, what are the steps, how it leads to increased synthesis of this enzyme, it is not known. And here all the enzymes, not only carbamoyl phosphate synthetase 1, but all of these 5 enzymes are stimulated. Now we have to look at the condition which leads to increased amino acids in the blood for long time. The first is when we take a high protein diet for the long time, high protein diet for the long time, then what does it leads to? It leads to increased amino acids for the long time, and which ultimately lead to increased synthesis of all enzymes of the urea cycle, and so there will be increased urea cycle, right? Now this is actually beneficial. See, if we are taking high protein diet for the long time, then this protein or amino acids must be catabolized, and that is being showed by increased urea cycle activity. The second condition which can lead to such increased amino acid for the long time is the fasting for a long time. Fasting for long time, or you can say prolonged starvation. Prolonged starvation. Now what happens in this prolonged starvation, that in the prolonged starvation, carbohydrate and fat as a fuel source, they are already depleted. So here in this, what happens, there will be increased breakdown of muscle protein. Increased breakdown of muscle protein. Muscle protein is broken down for the energy purpose, and because of breaking down of muscle protein, there will be again, it leads to increased amino acid for the long time, and which ultimately lead to increased urea cycle. So here I want to emphasize one more time on this, that this thing is the observation. What is the logic, or what is the sequence of events that lead to increased synthesis of all enzymes of urea cycle, that is not known. So that's all about regulation of urea cycle. Thank you.